Have you ever noticed the distinct coat colors of Labrador retrievers? You know, the classic black, the rich chocolate, or that sunshine
Oh
golden.
yeah, absolutely. They're all labs, but so different visually.
Right. It seems like it should be, I don't know, a straightforward genetic lottery, doesn't it?
You might think so.
But what if I told you that, well, while Mendel gave us the basic rules for how traits get passed down, nature, it always does this, adds some really surprising twists.
Inheritance isn't always simple.
Exactly, and those distinct lab variations, they're actually a perfect window into that complexity.
A great starting point.
Welcome to the Deep Dive, the show where we take your sources, we sort of peel back the layers, and really extract the most important bits of knowledge and insight.
Glad to be diving in.
Today, we're going deep into Chapter 4 of Essentials of Genetics, 10th edition. Specifically, the focus is modification of Mendelian ratios.
A really foundational chapter for understanding genetic complexity.
Totally. Our mission today is to go, well, way beyond the classic 3.1 and 9.3.3.1 ratios you might remember from, high school biology.
Yeah, those are just the beginning.
We're going to uncover how gene expression can be much more intricate than just simple dominant versus recessive.
We'll look at things like multiple genes influencing one trait,
How the environment can literally change what we see.
and even how DNA outside the nucleus plays a role. It's pretty fascinating stuff.
Get ready for some genuine aha moments because the world of genetics, it's way richer than just those basic Punnett squares.
Definitely.
Okay, let's unpack this.
Right. So to really appreciate these modifications, we first need to quickly remember Mendel's core ideas.
The fundamentals.
Exactly. Genes are on homologous chromosomes, right? And these chromosomes segregate when gametes form, and they assort independently.
Over the bedrock principles, yeah.
Yeah.
How genetic info gets transmitted.
All right.
But the text makes it clear these modified ratios start popping up when gene expression doesn't follow that simple dominant recessive pattern.
Or when more than one gene pair kind of works together to influence a single characteristic.
So it means genetics can be, well, a lot more intricate than those basic models suggest.
Precisely. And this complexity often starts with the alleles themselves.
Alleles, the different versions of a gene.
Yep, alternative forms of the same gene. We often talk about a wild-type allele. That's usually the most common or, normal version in a population. It's our baseline.
That it's often dominant.
Often, yeah, but not always. It just serves as the standard we compare mutations against.
Okay, so if the wild type is the standard, what happens when a gene mutates? How does that, show up in the organism?
Well, a mutant allele has modified genetic info. That usually leads to an altered gene product and ultimately a new phenotype.
And these mutations can affect how the gene works in different ways.
For sure. For instance, a loss-of-function mutation might reduce or even completely eliminate an enzyme's ability to do its job, like binding its substrate.
And if it completely stops working?
If the loss is total, we call that a null allele. Basically, it produces no functional product.
Okay, so it's not always about things breaking or becoming less efficient. Sometimes a mutation can actually, boost a function, maybe even dangerously so.
Exactly. That's where gain-of-function mutations come in. These can enhance the wild-type product's function, maybe by making more of it.
And these are usually dominant.
Generally, yes. A really significant example is proto-oncogenes. They normally help regulate cell cycles.
Right, keeping cell division in check.
But a gain-of-function mutation can turn them into oncogenes, which overrides that normal control.
Leading to cancer. That's a gain of function with huge consequences.
Definitely.
And the text also mentions neutral mutations. What are those?
Yeah. These are changes in the DNA sequence, but they don't cause any detectable change in the protein's function or the organism's phenotype.
So you wouldn't even know they're there unless you sequence the DNA.
Pretty much. They don't really affect the organism's fitness, so they just kind of, fly under the radar. Shows how much unseen variation exists.
Right. And it's also key, as the source points out, that traits are often influenced by multiple genes, not just one mutation.
Absolutely. Think about metabolic pathways. They're often long chains of enzymatic reactions.
It's the production line.
Exactly. A mutation in any gene coding for an enzyme in that pathway could stop the final product from being made.
So one observable trait, like the final product missing, could actually be caused by mutations in several different genes involved in the process.
Precisely. It highlights how interconnected everything is.
That makes sense. Now, thinking about all these allele types, I've noticed geneticists use a whole bunch of different symbols for them. Why so many?
That's a good question. It can seem a bit confusing at first. Mendel, of course, used the simple uppercase for dominant, lowercase for recessive, D for tall, D for dwarf.
That works well for simple
It
cases.
does, but then you have systems like the one used for Drosophila, the fruit flies.
Right, I've seen those.
They often use the initial letter or letters of the mutant trait name. It's lowercase if the mutant is recessive, uppercase if it's dominant. And the wild type gets a superscript plus light.
So like E for the ebony body mutation and E plus for the normal wild type gray body. It visually flags mutant versus standard.
Exactly. And then when you have cases where there's no clear dominance, like incomplete dominance or codominance, the symbols change again.
How so?
You often see uppercase italic letters with superscripts, like R1 and R2 for snapdragon flower color, or LM and LN for the MN blood group, or IA and IB for ABO blood types.
So that notation itself tells you, hey, neither of these is completely masking the other.
Precisely. It signals a different kind of relationship between the alleles.
And beyond those, the source mentions function-based symbols. Those seem really practical.
It can be very informative. Like in yeast, CDK stands for the cyclin-dependent kinase gene. Or in bacteria, Lu might indicate a mutation affecting leucine synthesis.
And in humans, things like BRCA1 for breast cancer susceptibility.
Right. These symbols tell you something about the gene's function or the trait it's linked to, which is useful in a different way. Each system is just a tool suited for describing different genetic situations.
Okay, so we've got simple dominant recessive. But what happens when alleles don't quite play by those rules? Let's start with incomplete dominance, sometimes called partial dominance. I think of it like mixing paint. You start with red and white, but instead of one color winning, you get pink.
That's a perfect analogy. Neither allele fully dominates, so the heterozygote has an intermediate phenotype, a blend.
The classic example is snapdragon flowers, right?
Exactly. Cross a pure red, R1R1, with a pure white, R2R2, and all the F1 offspring are pink, R1R2.
A perfect blend. And what happens if you cross those pink ones?
If you cross the F1 pinks, the F2 generation shows a 1.2.1 ratio, 14 red, 12 pink, and 14 white.
And what's neat is that the phenotype ratio, 1 red, 2 pink, 1 white, is exactly the same as the genotype ratio, 1R1R1, 2R1R2, and 1R2R2.
Precisely. That match immediately tells you it's likely a single gene with incomplete dominance at play.
That flower example is super clear. But can we see this kind of blending in humans too? Maybe less obviously.
Absolutely. Often it's clear at the biochemical level. Take Tay-Sachs disease. It's an autosomal recessive disorder.
Devastating disease, yeah.
Homozygous recessive individuals have almost no activity of a crucial enzyme, hexosaminidase A. But here's the incomplete dominance part. Heterozygotes, the carriers, are phenotypically normal. They don't have the disease.
Okay.
But if you measure their enzyme activity, they typically have about 50% of the normal level.
So it's intermediate at the enzyme level, even if they look healthy?
Exactly. It's enough for normal function, but biochemically, it's a clear intermediate state, not full wild-type activity, not the near-zero activity of the affected homozygote.
That makes a lot of sense. So incomplete dominance gives us a blend, but then there's codominance. People mix these up sometimes, right? How is codominance different?
It's a really common point of confusion, but the distinction is key. With codominance, you don't get a blend. Instead, both alleles are fully and clearly expressed in the heterozygote.
So both show up distinctly, not mixed.
Right. Think of it like maybe spots or patches of two different colors rather than a blended color. Both original phenotypes are present simultaneously and distinctly.
Okay. What's a good human example?
The MN blood group system, the classic one. There are two forms of a glycoprotein on red blood cells, M and N. They're determined by the LM and LN alleles.
So if you're LM-LM, you're type M blood. If you're LN-LN, you're type N.
Correct. And if you're heterozygous, LM-LN, you produce both the M and the N glycoproteins. Both are fully expressed and detectable on your red blood cells.
No blending to some intermediate protein, just both M and N fully present.
Exactly. That's codominance, distinct expression of both alleles.
That clarity helps a ton. Okay, so we've seen blending, we've seen both show up. Here's where it gets really interesting. What if there are more than just two versions of a gene out there in the population?
Right, that leads us to multiple alleles.
So even though any one person being deployed can only have two alleles for a gene.
Within the population as a whole, there can be many more different versions circulating. 3, 4, sometimes hundreds. The absolute classic example, and one with huge practical importance, is the ABO blood group system in humans.
Discovered by Carl Landsteiner way back when.
That's the one. It's controlled by three main alleles, IA, IB, and I. They're all on chromosome 9.
And the dominance relationship is interesting here, isn't it?
It is. IA and IB are both dominant over I. But, and this is key, IA and IB are codominant with each other.
Which explains the different blood types, right? Type A can be IAIA or IAI. Type B is IB or IA.
Exactly. And type O is 2, recessive. But if you inherit both IA and IB,
you get type AB because they're codominant, and both A and B antigens are expressed on the cells.
Precisely. And understanding this is just crucial for things like blood transfusions and organ transplants. Getting it wrong can be fatal. It's life or death genetics.
Absolutely. But then there's that really strange case, the Bombay phenotype.
I is a fascinating curveball. It's very rare, but some individuals appear to have type O blood, even if their ABO genotype includes IA or IB alleles.
So how does that happen? It's like the ABO genes aren't working.
It's actually due to a mutation in a completely different gene called FUT1. This gene makes something called the H substance.
And the H substance is needed for?
It's the precursor molecule that the A and B antigens normally attach to on the red blood cell surface. Think of it as the foundation.
So if you have a mutation in FUT1, specifically if you're homozygous recessive H, you can't make the H substance foundation.
Exactly. And without that H substance, the A and B antigens, even if the enzymes to make them are there, thanks to IA or IB alleles, have nothing to attach to. They can't be expressed on the cell surface.
So the blood tests it as type O because it lacks A, B, and H antigens, even if genetically the person should be A, B, or AB. Wow, that's a complete masking effect from another gene.
It is. What's fascinating here is how a gene at one locus, FUT1, can completely override the expression of genes at another locus, ABO. It's a beautiful example of gene interaction, specifically epistasis, which we'll probably get into more later.
Definitely seems like it. The source also mentions the white locus in fruit flies as another example of multiple alleles.
Oh yeah, Drosophila genetics is full of great examples. The white gene, which affects eye color, has over 100 known alleles.
100 versions of one gene.
Yep. And they result in a whole spectrum of eye colors from basically no pigment, white eyes, to deep ruby, orange, buff, all sorts of shades, but all with less pigment than the wild-type brick red eye. It really shows the potential for variation within a single gene.
Okay, so multiple alleles exist. Now, what about lethal alleles? That sounds pretty serious.
It is. This concept underscores that many gene products are absolutely vital for an organism just to survive.
So a mutation that messes up an essential function can actually be deadly.
Right. A mutation leading to a non-functional essential product can be lethal. It highlights that genes aren't just about, eye color or height, but fundamental life processes.
And there are different kinds, like recessive lethal alleles.
Yes. An individual can often tolerate one copy of a recessive lethal allele if they also have one normal wild-type copy. The heterozygote survives because the wild-type allele provides enough function.
But if they inherit 2 copies of that recessive lethal allele,
then the individual usually won't survive. The homozygous recessive genotype is lethal. The timing can vary. Death might occur during embryonic development or sometimes later.
So certain genotypes are just incompatible with life.
Exactly. A classic example is coat color in mice. There's a yellow coat color allele, AY, that's dominant for color over the normal agouti grayish color.
So heterozygous mice, AYA, are yellow.
Right. But that same AY allele is also a recessive lethal when homozygous. If a mouse inherits 2 copies, AYY, it dies before birth.
So if you cross 2 yellow mice, AYX, AYA, you don't get the expected 1.2.1 ratio among the offspring you actually see.
Correct. You'd expect 14 AYAY, lethal, 12 AYA, yellow, and 14 AYA, agouti. Since the AYA mice die in utero, the ratio among the live-born offspring becomes 2 yellow my 1 agouti.
So the lethal allele literally removes one expected genotype category from the observable progeny and changes the ratio to 2.1, or technically 23 yellow 0.13 agouti.
Precisely. It modifies the expected Mendelian outcome.
Wow. And then there are dominant lethal alleles, where just one copy is enough.
Yes. With these, the presence of just one copy of the lethal allele results in the death of the individual. Huntington disease in humans is the prime example.
Caused by a dominant allele.
Right. But the key thing with Huntington's is that the onset of the symptoms and the eventual lethality in heterozygous age is usually delayed until adulthood, often around age 40 or later.
Which is often after people have already had children.
Exactly. And that delayed onset is why the allele persists in the population, unfortunately. Affected individuals can pass it on before they even know they have it. It's a tragic pattern.
So, looking back at this section, incomplete dominance, codominance, multiple alleles, lethal alleles. All these discoveries really expanded the idea of what a gene is, didn't they?
Absolutely. It moved way beyond Mendel's simple unit factors.
Hmm.
It showed that alleles can affect the phenotype in really diverse ways, sometimes with complex or even deadly results.
And mutation is the engine driving all this variation, creating these new alleles.
That's the ultimate source, yes.
Okay, so far we've mostly focused on how single gene pairs can behave in these non-Mendelian ways. But what happens when we start combining these different modes of inheritance?
Now it gets even more interesting. Mendel's principle of independent assortment, that genes for different traits segregate independently, still generally holds if the genes are on different chromosomes or far apart on the same one.
Right, they aren't linked.
But if you combine different inheritance patterns in a dihybrid cross, looking at two traits at once, that classic 9.3.3.1 ratio you expect can get modified pretty significantly.
The book is an example, right? Combining albinism and ABO blood type.
Yes. Imagine a cross between two people who are both heterozygous for albinism, that's autosomal recessive, let's say A, and are also both blood type AB, which involves codominance, and multiple alleles, IAIB.
Okay, so you're tracking two traits: pigmentation, normal versus albino, and ABO blood type, AB, AB, or O.
Right. If you work out the combinations, maybe using a forked line method or a big Punnett square, you don't get the standard 4 phenotypes in a 9.3.3.1 ratio.
What do you get instead?
You actually end up with 6 distinct phenotypes.
6? Wow.
Yeah, you get normally pigmented people with type A, type AB, and type B blood, and you get albino people with type A, type AB, and type B blood. Yeah. The ratio works out to 3.6.3.1.2.1.
So 3 pigmented A, 6 pigmented AB, 3 pigmented B, 1 albino A, 2 albino AB, 1 albino B. That's definitely not 9.3.3.1.
Not at all. It really illustrates how combining different non-Mendelian inheritance patterns creates a much more complex landscape of potential outcomes.
And this leads to a broader point the chapter makes. Phenotypes are often affected by more than one gene. Gene interaction.
Exactly. Now, gene interaction doesn't always mean the gene products, like proteins, are physically bumping into each other.
Okay, what does it mean then?
It usually means that the functions of several different gene products contribute together to produce a single observable characteristic. It's like a team effort at the cellular level. Many proteins might be needed in a pathway to get to the final result.
The book mentions epigenesis here. Can you unpack that a bit?
Sure, epigenesis is more of a developmental biology concept, but it's relevant. It's the idea that development proceeds in sequential steps, and each step is under the control of one or more genes.
Like building something step by step, and different genes control each stage.
Pretty much. A great example is the development of the inner ear in mammals. It's incredibly complex.
Right.
Many genes are involved in that whole process. And a mutation in any one of those different genes can disrupt development and lead to the same outcome: hereditary deafness.
So deafness isn't caused by just one deafness gene.
Not at all. It's what we call a heterogeneous trait. In humans, mutations in over 50 different genes have been linked to hereditary deafness. One trait, many possible genetic causes.
That really highlights the interaction aspect. Okay, building on that, let's talk about epistasis. That sounds important.
It is. Epistasis is when the expression of one gene or gene pair actually masks or modifies the expression of a different gene or gene pair.
So one gene can basically interfere with or cover up the effect of another gene.
Exactly. The interaction can be antagonistic, like masking, or sometimes it can be complementary or cooperative, where genes need each other.
We actually already touched on an example with the Bombay phenotype, didn't we? That's recessive epistasis.
Perfect example. Being homozygous recessive at the FUT1 locus masks whatever ABO alleles you have. The recessive condition at one gene, FUT1, overrides the expression at the second gene, ABO.
And this connects back to our labs.
It does. Coat-colored Labrador retrievers, black, chocolate, and yellow, golden, is a classic example of recessive epistas.
How does that work?
Okay, so there's one gene, let's call it the B gene, that determines the type of pigment. B for black, dominant, and B for brown, recessive.
So BB or B would be black and B would be brown.
Right, but there's another gene, let's call it E, that controls whether pigment gets deposited in the fur at all. The dominant E allele allows pigment deposition, but if a dog is homozygous recessive, E.
Then no pigment gets put into the fur, regardless of the B genotype.
Exactly, so an E dog will be yellow, golden, whether it has the genes for black, B or brown B pigment. The E genotype is epistatic to the B gene. It masks the effect.
So that's how you get yellow labs. They might genetically be black or brown, but the E prevents that color from showing up. And this leads to a specific ratio in crosses, right?
It does. If you cross two dogs that are doubly heterozygous,
Mm-hmm.
BB, these are the black labs, the offspring ratio isn't 9.3.3.1. It comes out as 9 black, 3 brown by 4.
Ah, the 9.3.4 ratio. That 4 includes both the B and by B genotypes, all appearing yellow.
Precisely. That ratio is a hallmark of recessive epistasis.
Okay, so that's recessive epistasis. What about dominant epistasis?
Here, it's a dominant allele at one locus that masks the expression of alleles at a second locus. Summer squash fruit color is a good example.
Swash color.
Okay, there's a dominant allele, let's say W, that results in white fruit color. If a squash has even one W allele, it's white, regardless of the genotype at a second locus, say Y, which controls yellow versus green.
So W just overrides everything else.
Pretty much. Only if the squash is homozygous recessive W and A can the Y gene show its effect. Then let's say Y gives yellow fruit and Y gives green fruit.
So W is white, WWY is yellow, and WY is green.
Exactly. And if you cross two double heterozygotes, WWYY, which would be white, you get a 12.3.1 ratio in the offspring.
12 white, 3 yellow, 0.1 green. Makes sense because the dominant W masks in 12 out of the 16 combinations.
Correct. That 12.3.1 ratio signals dominant epistasis.
Interesting. Then there's complementary gene interaction. What's that about?
This is where you need at least one dominant allele at each of two different gene pairs to get a particular phenotype. They complement each other.
Both are required, like needing 2 different keys to open a lock.
Good analogy. Sweet pea flower color is the classic example. There are 2 different genes involved, let's say AA and B.
Yeah.
You need at least one dominant A and at least one dominant B to produce purple flowers.
So only the AB genotype gives purple.
Right. All other combinations, AB, AB, and AB, result in white flowers because one or both necessary components for the purple pigment pathway are missing.
So if you cross 2 true breeding white varieties, like ABXABB,
all the F1 offspring are AB, and they are all purple because they have one dominant allele from each gene.
But then in the F2 generation, when you cross the AB plants,
you get a 9.7 ratio.
Yeah.
916 are AB, purple, and the other 716 A, combined are white.
The 9.7 ratio signals that complementary action. Got it.
And one more type of interaction. Sometimes it yields entirely novel phenotypes that weren't seen in the parents or F1.
New shapes or colors appearing?
Exactly. Some are squash shape this time. If you cross a true breeding disc-shaped squash, say AAB, with a true breeding long-shaped one, the F1AB, are all disc-shaped.
Okay, so disc seems dominant.
It seems that way initially. But in the F2 generation from crossing ABB, XAB, you get 3 shapes. Disc, sphere, and long in a 9.6.1 ratio.
Wait, sphere, where did that come from?
That's the novel phenotype. It turns out that having a dominant allele at either locus AB or AB results in a sphere shape. The double dominant AB gives disc, and the double recessive gives long.
So 916 disc, AB, 6 octospheres, AB plus AB, and 116 long, AB. Wow, gene interaction creating something completely new.
It really shows the creative potential of combining gene effects.
So the big takeaway from all these modified ratios, you mentioned the numbers 9.3.4, 12.3.1, 9.7, 9.6.1.
Right. When you're studying a single characteristic and you do a cross that should give a 9.3.3.1 ratio if 2 genes were acting independently in a simple Mendelian way, but instead you see one of these ratios expressed in 16ths.
That's a huge clue, isn't it?
It's a very strong indicator that 2 gene pairs are interacting through epistasis, complementary action, or something similar to control that phenotype. It's your first hint that the genetics are more complex.
If we connect this to the bigger picture, understanding these modified ratios is really critical, isn't it?
Oh, absolutely. It's how geneticists can start to figure out gene interactions just by observing inheritance patterns, even before they know the specific genes or biochemical pathways involved. It's a powerful deductive tool for unraveling genetic complexity.
Beyond these interactions, there are even more layers affecting how genes ultimately show up, right? Like pleiotropy and how we figure out if mutations are in the same gene.
Yes, let's get into complementation analysis. This is a really clever experimental approach.
What's its main purpose?
Its purpose is to figure out if 2 different mutations that cause a similar phenotype, maybe you've isolated 2 different strains of wingless fruit flies, for example, are actually mutations in the same gene, or if there are mutations in 2 different genes that just happen to affect wing development.
So are they different alleles of one gene or mutations in separate genes altogether?
Exactly. The method is pretty simple conceptually. You cross the two true breeding mutant strains together.
And then look at their kids, the F1 generation.
Right. Now, there are two possible outcomes.
Okay.
Case 1. All the F1 offspring develop normal wings.
Okay, what does that mean?
That means complementation occurred. The mutations must be in separate genes. Each parent strain provided a normal functional copy of the gene that was mutated in the other strain.
So the mutations complement each other, restoring the wild type phenotype. Like one parent gives a good copy of gene A, the other gives a good copy of gene B, and the offspring gets both functional genes needed for wings.
Perfect analogy. Now, case 2. You cross the two wingless strains, and all the F1 offspring also fail to develop wings.
So no normal wings appear.
Right. This means no complementation occurred. The two mutations must affect the same gene. They are different alleles of that one wing development gene. Since the offspring didn't get a functional copy of that crucial gene from either parent, they remain wingless.
So this test lets you group mutations. All mutations that fail to complement each other belong to the same complementation group, meaning they're alleles of the same gene.
Exactly. It helps geneticists determine how many different genes are fundamentally involved in producing a particular trait, like wing formation.
Okay, that's complementation. Now, what about pleiotropy? We talked about multiple genes affecting one trait. This is the flip side.
It is indeed. Pleiotropy is when the expression of a single gene leads to multiple, often seemingly unrelated, phenotypic effects.
One gene having many different jobs or consequences?
Precisely. It's like one domino falling and triggering several different chains of events. You often see this when a gene product, like a protein, is used in different tissues or pathways.
The book gives Marfan syndrome as a human example.
Yes, a classic example of pleiotropy. It's an autosomal dominant disorder caused by a mutation in the gene coding for fibrillin.
And fibrillin is a connective tissue protein.
Right. And because connective tissue is found all over the body, a defect in fibrillin causes a whole suite of symptoms. Problems with the lens in the eye, increased risk of the aorta rupturing, lengthened long bones in the arms and legs.
What's stemming from that one faulty gene product? It's even thought Abraham Lincoln might have had it.
That's a common historical speculation, yes, based on his physical appearance. It shows how widespread the effects can be.
Another one mentioned is porphyria variegata.
Also autosomal dominant. This one affects the metabolism of porphyrins, which are components of hemoglobin.
And the symptoms are diverse.
Extremely. The buildup of porphyrins is toxic, leading to things like deep red urine, severe abdominal pain, muscle weakness, fever, rapid pulse, insomnia, headaches, vision problems, even delirium and convulsions.
Wow. Believed to affect King George III, it's hard to even categorize that as one thing.
Exactly. It highlights how a single biochemical defect can cascade through multiple physiological systems. Careful observation often reveals pleiotropy is more common than we might initially think.
Okay, so single genes can have multiple effects. But we also need to consider the environment, right? And the overall genetic background.
Absolutely critical. Gene expression and the resulting phenotype are almost always modified by interactions between the genotype and the organism's internal and external environment. It's rarely just about the gene itself.
Nature and nurture interacting. This brings us to concepts like penetrance and expressivity. Let's start with penetrance.
Penetrance refers to the percentage of individuals who have a specific genotype and actually show any degree of the associated phenotype.
So it's like, if you have the gene, do you show the trait at all? Yes or no?
Pretty much. For example, if a particular mutant gene is present in 100 flies, but only 85 of them actually display the mutant phenotype and 15 look wild type, we'd say that mutant gene has 85% penetrance.
So some individuals can carry the gene but not express it visibly.
Correct. Then there's expressivity. This deals with the range or degree of expression among individuals who do show the phenotype.
So if you show the trait, how much do you show it?
Exactly. The classic example is the recessive eyeless gene in Drosophila. Flies that are homozygous for this gene all have the genotype, but their phenotype can range dramatically.
How so?
Some might have completely normal eyes, others might have slightly smaller eyes, some might be missing one eye entirely, some might be missing both eyes.
Same genotype, but a huge range in how severely it's expressed. That's variable expressivity. And environmental factors can directly cause these variations sometimes, right? Conditional mutations?
Yes. Temperature is a very common environmental factor that affects phenotype. Chemical reactions, including enzyme activity, are temperature dependent.
Like the evening primrose example.
Right. Red flowers at 23 degrees Celsius, but white flowers at 18 degrees Celsius. The enzyme making the red pigment just doesn't work well at the cooler temperature.
And Siamese cats or Himalayan rabbits, that pattern is amazing.
It is. They have dark fur only on the cooler parts of their bodies. Nose, ears, paws, tail.
Because the enzyme that produces the dark pigment is temperature sensitive.
Exactly. It's only functional at the lower temperatures found in the extremities. The core body temperature is too warm, so the fur there stays light. It's a visible map of body temperature affecting gene expression.
And nutrition can play a role too.
Yes, nutrient availability can certainly influence how genes are expressed and what phenotypes result. And there are lab examples of temperature-sensitive mutations, where a mutant organism might show the mutant phenotype at one temperature, the restrictive temperature, but look completely normal, wild type, at another temperature, the permissive temperature.
Beyond the external environment, what about the internal genetic background, like position effects?
Yeah. Position effect is fascinating. It means the physical location of a gene on the chromosome relative to other genetic material can influence how actively it's expressed.
So where the gene sits matters.
It can. The example often used is the white gene, W+, for red eyes in Drosophila. If, through a chromosome rearrangement like a translocation, the normal W+ allele gets moved right next to a region of heterochromatin.
Heterochromatin being that tightly packed, generally inactive DNA.
Correct. When the active W+ gene is placed near this silencing environment, its expression can become unstable or repressed in some cells,
Leading to
variegated eyes, eyes that are mottled with patches of red, where W+ is expressed, and white, where it's silenced by the nearby heterochromatin. The gene itself is normal, but its neighborhood is affecting its expression.
Wow. Okay, another factor is the onset of genetic expression. Traits don't always show up at birth.
Very true. Many genetic traits, especially disorders, have a characteristic age of onset. This is often tied to the developmental stage when the gene product is most needed.
We mentioned some earlier. Tay-Sachs in infancy.
Lesch-Nyhan syndrome, around 6 to 8 months. Duchenne muscular dystrophy, typically diagnosed around 3 to 5 years.
And Huntington's much later, around middle age.
Right. The gene is present from conception, but its phenotypic consequences might not become apparent until much later in the organism's lifespan.
And finally, under these complexities, there's genetic anticipation. What's that?
Genetic anticipation is a pattern observed in certain heritable disorders, where the symptoms tend to become more severe and appear at an earlier age in successive generations within a family.
So kids might have it worse and get it earlier than their affected parent did.
Generally, yes. Myotonic dystrophy, an autosomal dominant muscle disorder, is a key example. The severity often increases, and the age of onset decreases as it passes down through generations.
What causes this anticipation effect?
It's often linked to the molecular nature of the mutation itself. In myotonic dystrophy and several other disorders like Fragile X syndrome and Huntington's again, the mutation involves an unstable expansion of short, repeated DNA sequences, trinucleotides, within the gene.
So the number of repeats gets bigger.
Yes, the repeat sequence tends to expand further when it's transmitted from parent to child. A normal gene might have, say, 5 repeats. Mildly affected individuals might have 50. Severely affected individuals, often with early onset, might have over 1,000 repeats. The larger the expansion, the more severe and earlier the disease.
It's like the mutation itself gets worse over generations.
In a sense, yes. It's a dynamic mutation.
So considering all this penetrance, expressivity, environment, position effects, onset timing, anticipation, it really makes you think, doesn't it?
It absolutely does. It raises that fundamental question. How much of what we observe is purely dictated by the genes, and how much is shaped by this incredibly complex interplay between our genes, our internal biology, and the external world around us?
It's clearly far more nuanced than a simple gene for X model often implies.
Much, much more nuanced.
And believe it or not, we're still not done. We have to go beyond the nucleus and talk about extranuclear inheritance.
Right. Inheritance patterns that don't follow the rules for genes located on the chromosomes in the nucleus.
This involves DNA found elsewhere in the cell.
Primarily, yes. There are 2 main types discussed here. The first is organelle heredity.
Meaning heredity determined by genes found in organelles like mitochondria and chloroplasts.
Exactly. These organelles contain their own small chromosomes, their own DNA. Phenotypes influenced by genes on this organelle or DNA often show patterns different from nuclear inheritance.
And a key feature is maternal transmission.
Often, yes, especially with mitochondria. Because the egg cell contributes the vast majority of the cytoplasm, including organelles, to the zygote, while the sperm contributes very little cytoplasm, these traits are typically inherited from the mother.
So if you do reciprocal crosses, swap which parent provides the egg, you get different results.
That's a classic sign of cytoplasmic or organelle inheritance. Carl Correns saw this way back in 1908 with 4 o'clock plants.
The variegated leaves.
Yes, Mirabilis callapa can have branches with all green leaves, all white leaves, or variegated patched green and white leaves. Correns found that the phenotype of the offspring's leaves was determined solely by the phenotype of the branch from which the ovule egg came.
The pollen source branch phenotype didn't matter at all.
Not at all. If the ovule came from a white branch, the offspring were white. If from a green branch, offspring were green. If from a variegated branch, offspring could be green, white, or variegated. It was purely maternal inheritance.
Indicating the genetic factor was in the cytoplasm, specifically in the chloroplasts, which determine leaf color. The white leaves result from a mutation in chloroplast DNA.
Precisely. And we see similar things with mitochondrial mutations.
Like Poki and Neurospora.
Right, the bread mold.
Yeah.
Poki is a slow-growing mutant strain because its mitochondria don't function properly for energy production. Crosses showed it's inherited through the cytoplasm maternally.
And Petite in yeast.
Saccharomyces yeast. Petite mutants form small colonies because they have defects in cellular respiration, again often linked to mitochondrial problems. Most petite mutations show cytoplasmic transmission, indicating they're in the mitochondrial DNA, mtDNA.
Though some petite mutations are actually in nuclear genes, right? Showing that both genomes have to work together.
Good point. Mitochondrial function relies on proteins encoded by both nuclear DNA and mtDNA. They have to cooperate.
So what about human mitochondrial genetics?
It's a huge field now. We know the entire sequence of human mtDNA is about 16,569 base pairs. It's small but mighty.
What does it code for?
It encodes 13 essential proteins involved in aerobic respiration, the electron transport chain, plus 22 transfer RNAs and 2 ribosomal RNAs needed for making those proteins inside the mitochondria.
So mitochondria have their own protein synthesis machinery.
They do. But, and this is crucial for disease, mtDNA seems to be more vulnerable to mutations than nuclear DNA.
Why is that?
2 main reasons are thought to be important. 1, the repair mechanisms for mtDNA damage seem less efficient than those for nuclear DNA. 2, mitochondria are the site of respiration, which generates a lot of reactive oxygen species free radicals that can damage DNA.
So they're in a high-risk environment with less protection.
Kinda, yeah. And mutations in mtDNA can cause a range of human disorders, often affecting tissues with high energy demands, like muscle and nerve tissue.
MRF was mentioned as an example, myoclonic epilepsy and ragged red fiber disease.
Yes. It clearly shows maternal inheritance only offspring of affected mothers get it. Symptoms include muscle weakness, ataxia, deafness, dementia, seizures.
And the ragged red fibers.
That refers to a finding in muscle biopsies, where you see clumps of abnormal, proliferating mitochondria staining red. It's caused by a mutation in a mitochondrial gene for a specific transfer RNA, tRNAs.
And the idea of heteroplasmy is important here too.
Very important. It means that within the cells of an affected individual, there's usually a mixture of mitochondria, some with a mutation, some normal.
So not every mitochondrion is mutant.
Right. The proportion of mutant mtDNA versus normal mtDNA can vary between tissues and individuals, and this often influences the severity of the disease. If you have mostly normal mitochondria, symptoms might be mild or absent. If you have a high percentage of mutant mtDNA, the disease is usually more severe. It dilutes the effect.
And mitochondrial dysfunction is now linked to lots of common diseases and maybe even aging.
There's a lot of research suggesting that. Problems in mitochondria are implicated in everything from anemia and blindness to type 2 diabetes, Parkinson's, Alzheimer's, even some cancers. And the idea that accumulating mtDNA damage over our lifetime contributes to the aging process is a major hypothesis.
Which leads into that really cutting-edge and sometimes controversial area, mitochondrial replacement therapy.
Right, or MRTs. These are techniques developed to try and prevent mothers who carry known pathogenic mtDNA mutations from passing them on to their children.
Because these diseases can be so devastating.
Exactly. Cytoplasmic transfer was tried earlier but had issues. MRTs are more refined. The 2 main methods are maternal spindle transfer, MST, and pronuclear transfer, PNT.
Can you briefly explain those?
Sure. With MST, you take the patient's egg, which has the unhealthy mitochondria but the mother's nuclear DNA on the matic spindle. You carefully remove that spindle and transfer it into a donor egg that has had its own nucleus removed but contains healthy mitochondria. Then you fertilize that reconstructed egg in vitro.
So mother's nuclear DNA, donor's healthy mitochondria. What about PNT?
With PNT, you fertilize both the patient's egg and a donor egg in vitro separately. Before the pronuclei, the sperm and egg nuclei fuse, you transfer the pronuclei from the patient's zygote into the donor zygote, which has had its own pronuclei removed.
Again, resulting in an embryo with nuclear DNA from the parents, but mtDNA from the donor.
Correct. In both cases, the aim is to produce a child free from the mother's mitochondrial disease.
But this leads to the three-parent babies label in the media.
It does, because technically there's genetic material from 3 individuals, nuclear DNA from mother and father, and mtDNA, a tiny fraction of total DNA, but still distinct from the donor.
And that sparks ethical debates, right? About germline modification, identity.
Huge debates. Is it ethical to modify the human germline in this way, even to prevent disease? What are the long-term consequences? Does having DNA from 3 people affect identity? These are complex societal and ethical questions that science alone can't answer.
Okay, that's organelle heredity and MRT. The other type of extranuclear inheritance mentioned is maternal effect or maternal influence. How is this different?
This is different because the genes involved are actually in the mother's nucleus. But their products, proteins or messenger RNAs, are deposited into the egg cytoplasm before fertilization.
So the mother loads up the egg with instructions.
Exactly. And these maternal gene products in the egg cytoplasm direct the very early stages of embryonic development after fertilization, sometimes regardless of the embryo's own genotype for those genes.
So the offspring's early phenotype is determined by the mother's genotype, not its own.
For certain traits controlled by maternal effect genes, yes. The classic example is the bicoid gene in Drosophila development.
What does bicoid do?
It's a nuclear gene in the mother that produces a protein crucial for establishing the anterior-posterior head-to-tail axis in the embryo. The mother deposits bicoid mRNA at the anterior pole of her egg.
Okay.
Now, if the mother fly is homozygous recessive for a non-functional bicoid allele, BCD to ACD, she cannot deposit functional bicoid mRNA into her eggs.
So her eggs lack the instruction for make a head end.
Right, and consequently, embryos developing from these eggs fail to develop anterior structures like the head and thorax. They basically develop 2 tail ends.
And this happens even if the embryo inherits a normal functional BCD plus allele from the father.
Yes. Even if the embryo's own genotype is BCD plus BCD, it still shows the mutant phenotype, no head, because the crucial bicoid product needed very early on had to come from the mother's contribution to the egg cytoplasm, and it wasn't there.
Wow. So the mother's genotype dictates the offspring's phenotype for that early developmental step. That's a powerful influence.
It really highlights how the oocyte cytoplasm, prepared by the mother, provides critical information for kicking off development.
Okay, so let's try and wrap all this up. We've covered a lot.
We certainly have.
This deep dive really shows that while Mendel's basic principles of how genes are transmitted still hold true,
the way those genes are expressed and interact is incredibly complex and varied.
We've seen incomplete dominance, codominance, multiple alleles, lethal alleles messing with ratios.
Complex gene interactions like epistasis and complementary action creating unexpected phenotypes.
The fact that one gene can have multiple effects, pleiotropy.
How the environment, things like temperature or even a gene's location, can change expression, penetrance, expressivity, position effects.
Not to mention things kicking in at different times or even getting worse over generations like in anticipation.
And then topping it all off with inheritance patterns totally outside the nucleus, like mitochondrial DNA and maternal effects.
It paints a picture of heredity that's vastly more intricate than just simple dominant recessive Punnett squares.
Absolutely. It's a dynamic interplay of many factors.
And if we connect this to the bigger picture, understanding these nuances is just crucial, isn't it?
Oh, completely. It's fundamental for everything from breeding more productive crops or healthier livestock, to understanding, diagnosing, and potentially treating human genetic disorders. It forces us to move beyond simplistic categories.
We have to appreciate the complexity to make progress.
Exactly. It challenges us to think about the intricate biological dance that creates all the diversity we see in life.
So, what does this all mean for you, our curious listener?
Well, I think it means the world of genetics is just far richer, more interconnected, more fascinating than maybe we initially imagined. Every single trait, every variation we see is really a testament to this incredibly intricate dance of genes, gene products, interactions, and environmental influences.
It's definitely not simple cause and effect most of the time. So here's a final thought to mull over. Considering how deeply genes, the environment, and even those initial conditions set up in the egg by the mother can influence development. What kind of revolutionary breakthroughs might we see in the future if we learn to better understand or maybe even manipulate these complex interactions?
And importantly, what profound ethical questions will that inevitably raise as our capabilities grow?
Lots to think about there. Thank you so much for joining us on this deep dive into the fascinating modifications of Mendelian ratios.
We hope you found it insightful.
Yeah, we hope you gained some surprising insights and maybe feel a little more informed about the truly incredible complexity of life and heredity.